Hi, welcome to the first presentation on cell division, diversity and cellular organisation. Today I'm going to focus on what we call the cell cycle and the process of mitosis, hopefully some of which you might already be familiar with from GCSE. So just looking at the specification, we're going to be looking today at the cell cycle, how the cell cycle is regulated, what the main stages of mitosis are, a brief look at a section of plant tissue showing the cell cycle and stage of mitosis, or at least mitosis stages, and why mitosis is significant or important. Let's begin with what the cell cycle is and how it's regulated or controlled. So, what's the point of the cell cycle? Well, we need to make, all organisms need to make new cells, and they can only do that by a process what we call cell division. And obviously the main processes of cell division are mitosis and meiosis. Remember that mitosis is for regular cells, meiosis only for the production of sex cells. So all of the cell division is mitosis. Meiosis is the only type of cell division where we're producing gametes. So eggs and sperm, or pollen and ovules in plants as well can be those. Obviously different cells can divide in different ways. There's lots of reasons why things might wish to be replaced. For example, you need to replace damaged cells, cells that get brushed and knocked a lot, cells that are growing through things. So intestinal cells where food is pushing past them, that is required to be replaced every 5 days. So here we've got some epithelial cells. Some cells only divide for a specific purpose, like liver cells, bone marrow cells to produce new bone, and what we call the meristem, that's found in plants. So at the tip of a root or the tip of a shoot, so in other words where it's undergoing cell division, and the cambium where it's dividing to make new xylem and phloem cells. As we said, mitosis and meiosis. Mitosis for growth and repair, meiosis sex cells only. Now remember that in prokaryotes or bacteria we don't have mitosis or meiosis, we do have a cell division process, but it's known as binary fission. It's not mitosis or meiosis because they are nuclear cell division, and obviously bacteria don't have a nucleus. They don't have a membrane-bound nucleus, they just have a loop of DNA. So, but the principle behind the process is similar. There's duplication of DNA, then splits into two separate cells, and then divides into those two separate cells. So it's a similar kind of process, but without a nuclear membrane involved. So what we call the cell cycle is the whole process from start to end. So in other words, from when a new cell first develops to when it completely divides to produce two new cells. And cells spend a lot of their life cycle or their cell cycle in what we call interphase. In other words, in between cell division. Yeah, only a small amount of time is spent in actual cell division itself, or mitosis. So interphase, the sort of key parts of the cell cycle, it's divided into several sections known as G1, S and G2. We'll look at those in more detail in a moment. But in interphase, it's where cells grow, where cells are preparing themselves for cell division. For example, they're making new DNA, they've got to replicate their DNA, make a copy of it. They've got to replicate all the organelles for the new cells. So a lot of activity is happening, although it's not visible under a light microscope. It's very difficult to see because it's quite small. Yeah. Mitosis, however, is easy to identify, and we'll look at the appearance of cells undergoing mitosis shortly. Now, the important thing to remember about mitosis is not only is it producing two new, what we call daughter cells, always classed as daughters from the original parent, but that you must say that they are genetically identical. Not just they're identical, but you must use the word genetically identical in your definition. If you don't say genetically, you will lose a mark for on mark schemes. Yeah, so make sure you're picking up on the idea that they are genetically identical cells. So let's look at the cell cycle in more detail. G1, the first stage of what we're calling the cell cycle. G for growth, it's the longest part of the cell cycle, the longest phase. It's when protein synthesis is taking place, so to produce new organelles. More cytoplasm, the cytoplasm grows in size. Cells will differentiate, so in other words, they become adapted to become a certain type of cell, which involves switching on different genes so that they can become that type of cell. And the length of time they spend in G1 phase will entirely depend on which type of cell it is. And cells can sometimes remain in G1 or interphase permanently. And at this point, the DNA is not yet replicated. So, for example, if there were 20 chromosomes, there would be only 20 chromosomes. In the S phase, the next phase which follows on from G1, that's when the DNA replicates. And this must occur if cell division is to occur. You've got to have enough DNA for each nucleus in the new cell to have the same amount of DNA as the old cell. So you've got to replicate the DNA as part of the process. And then G2, another shorter growth phase. It's where you get the preparation for mitosis. So the cell's cytoskeleton breaks down, and the components are used to make what we call the spindle fibers, or are ready to produce some spindle fibers. Remember, there's still twice the amount of DNA that it used to be because it's getting ready to split that DNA up in the process of mitosis. We'll look at the individual phases in a moment. And then you have something called cytokinesis. We'll look at that in more detail, but basically that's when the cell actually splits into two. So let's go on to mitosis itself. Why do we need mitosis? It's for growth, repair, replacing worn out cells, and it's an extremely similar process for things which are reproducing asexually. So if we need to increase the number of cells, or when an organism is growing and growing bigger, and then it undergoes mitosis. And as I said, the key thing to say about mitosis is it's making genetically identical cells. So it's copying and replicating and making an identical copy. Why do we need mitosis to be controlled? Well, if it's not controlled, we end up with beautiful things like this, where we end up with tumours. Tumours are uncontrolled cell division by mitosis. So we've got a failure of the cell cycle to be controlled and regulated. You get rapid and doubling and replication of cells. And this is obviously not what we want. So we want the cell cycle to be controlled and replicated. Now, to understand mitosis, it's a good idea to review our understanding of what we call a chromosome. Chromosome literally means coloured body. And chromosomes are made up of chromatin. Chromatin is a combination of DNA and histone proteins. DNA is coiled or looped around histones, and that makes chromatin altogether. Now, before we get the replication, before we get the cell division, we get replication. So the single chromosome then divides to make two what we call sister chromatids, so two identical sets of the same information held together in the middle by something called a centromere. So you get this classic X sort of shape. The middle bit is the centromere. Each one of those is a chromatid. And it coils up so we can actually see them. We can see chromosomes undergoing cell division with a light microscope. You can't normally see chromosomes otherwise. So let's just whiz through this bit and skip to where mitosis might occur. Again, we've mentioned this already, where we need to replace cells, skin cells, intestinal lining cells, red blood cells, which wear out quite quickly because they don't have a nucleus, where tissues are growing rapidly. For example, think about a zygote or a ball of cells that's reproducing. Each cell needs to replicate and replicate and replicate until it becomes a full-sized organism. Or in the immune response, where we need to rapidly produce lots of cells to fight off a disease, for example. Yeah. Okay, here we go. It's time to think about the different stages of mitosis. So it begins with prophase. We've got the DNA that's been copied in interphase. The chromosomes condense and become shorter and then become visible. The outside of the nucleus, the nuclear envelope breaks down so that the chromosomes are just found in the cytoplasm. And we have something called a centriole, these little T-shaped things here, which move to opposite ends of the cell, forming the spindle across them as they do so. So these are fibres which sort of run the length of the cell. Second phase is what we call metaphase. The chromosomes line up along the midline of the cell, which we're calling the equator. And the spindle fibres then move to the opposite ends, which we're calling the pole. And they were pulled along by their centromeres. The centromeres actually line up on the spindle fibres and move along the spindle. So as the spindle fibres pull each of the centromeres into anaphase, the two separate chromatids get split up. You get one going to each end, and that requires energy. The moving of the chromatids to each end, that's the bit of this process that requires energy to take place. And the final stage is telophase. So you end up with the chromatids going back to the opposite poles, uncoiling, disappearing, called chromosomes again. You stop seeing them with the light microscope, and then as the spindle breaks down, you get a nuclear envelope forming around each one, so forming two nuclei. And it's followed by something called cytokinesis, which we'll talk about in a moment. But you need to remember PMAT, P-M-A-T, prophase, metaphase, anaphase, telophase, and an idea of what the chromosomes are doing in each one. A quick representation, both in diagrammatic form, and also you need to be familiar with the light microscope form for each one. So prophase, you can just about see the sort of separate chromosomes here. Metaphase, classic, lined up on the equator. Anaphase, they're just starting to pull apart here. You can see them pulling apart and separating here. And, hang on. We skipped telophase, which is here, and it's just starting to become two separate cells. So, cytokinesis, slightly different in animal and plant cells. In animal cells, you get a waist forming, so in other words, like a narrowing, a bit like an old-fashioned Victorian corset. The two cells gradually narrow and narrow and narrow until you get two new cells, and that's caused by constriction or contracting of proteins. In plant cells, it's slightly different. There we go. You get the Golgi and the vesicles starting to form something called a cell plate. So they line up in the midline between the two and form a cell plate. You have still retained some connections, whereas in animal cells they're completely separate. In plant cells you get some connections between the two. So cytoplasm actually runs through these things called plasmodesmata. Here's an appearance of a cell under sort of a meristem, what we call the root tip from a plant. So this is probably an onion or garlic, are classic ones used for this. This is the bit which is kind of ready to be sloughed off. Often it gets dragged off because it's having to push its way through the soil. But the bit where it's rapidly undergoing mitosis is here, where we get these sort of more rectangular or cuboidal shaped cells. Obviously you can't see the individual chromosomes here, but in practicals that's the bit you're going to be looking for when you're looking for the sort of evidence of mitosis. It's underneath the root cap, so you can see the curved bit of the root cap, and it's just a little bit beyond or behind that, and you get these regularly repeating cells where you can see lots of nuclei. Okay, I'll post this link to the bottom of or underneath the video. It's a good sort of clip on the stages of mitosis. Obviously I've shown you some pictures, but it's quite useful to see sort of an overview of the actual stages of the mitosis itself. Okay, please make sure you check through this again. Make sure you can remember all the key stages of mitosis and of the cell cycle and what happens in each one. Okay, happy learning.